Hello students, our today's topic is growth and development. So first of all, you should know what is growth. The growth refers to quantitative. Growth is quantitative. Increase in size, mass or the volume. According to Todd, growth is increase in size. According to Profit, growth is increase in the size and the number. What is development? Development is a progress toward the maturity. Now, what is difference between growth and development? Growth, it is a quantitative increase in cell size, mass or volume, whereas the development, it refers to the qualitative abilities or progression in skills or abilities. In the context of living organism, growth refers to increase in the physical size or the mass, as in case of a plant that is growing taller or a child that is gaining a weight. On other hand, development refers to maturation of physical, mental or emotional capacity, such as child that is learning to walk or person developing the social skills. Next are the growth spurts. So growth spurts, they are the period of the rapid growth in height and weight. So following are the timings of the growth spurt. Just before the birth, the first growth spurt is seen just before the birth and 1 year after the birth. Other is seen in the mixed dentition growth spurt, that is seen in boys at the age of 8 to 11 years and girls at the age of 9 years. The prepubertal growth spurt, it is seen in boys at age of 14 to 16 years and girls at the 11 to 13 years. So remember, the first growth spurt typically occur during the infancy and early childhood, with the most rapid growth occurring in the first year of the decade. The maximum growth occur in the first year of life. So there is the babies, they can grow at average of 10 inches and triple their birth weight. After the first year, the growth, it tends to slow down. So these growth spurt will be dependent on the various factors like the nutrition, overall health and genetics. Some children may experience earlier or some may experience the late growth spurt. Other important growth curve that you should remember for your exam is what is Squamon growth curve. It is a graphical representation of the stages of growth and development in the humans. So the Squamon growth curve, it provides a framework for understanding the stages of growth and development in the humans. According to Squamon, he noticed the growth in the number one is the lymphoid tissue. The lymphoid tissue, they include the lymph nodes, spleen, thymus, tonsils and other lymphoid organs. So the lymphoid organs, they reaches a growth spurt of around 200% of their adult size by the age of 18 years. And a neural tissue, they reaches to their full size and shape by 6 to 7 years. So remember, during childhood and adolescent, the lymphoid tissue, they grow rapidly, so reaching a peak around the 18 years. So what happens after 18 years? After this age, the lymphoid tissue, it begins to decrease in size and activity. So although it remains important for maintaining the immune function throughout the adulthood. The neural tissue that will include the spinal cord, brain, and they are responsible for controlling all the bodily functions and the activity. So neural tissue, it grows rapidly during the first few years of life, reaching the full size at 6 to 7 years. That is what you have to remember for your exam. It reaches its full size and shape by age of 6 to 7 years. So after this age, the neural tissue, it continue to develop and mature. So overall, the size of shape of brain and nervous system, it doesn't changes significantly. Other are the genital tissue. The genital tissue, it grows at a slower pace during first 10 years. However, after the pubertal growth spurt, the genital tissue growth accelerates rapidly and it reaches maximum growth by age of 16 to 18 years. The last are the general tissue. The general tissue, they grow at a slower pace during the first few years of life and reaching about 50% of adult size by age of 2 to 3 years. So the general tissue, they includes the tissue system that gives shape and support to body, including bone, muscles, cartilage, connective tissue, etc. So it is the Scamone curve of the growth. Other important thing is cephalocaudal growth pattern. What is that? According to the cephalocaudal growth pattern, the growth and development occurs in most organisms, in which growth and development it occurs in head to the tail direction. So the growth, it will begin at the head part and gradually progress downward toward the feet. So during the prenatal, what you will see, the growth pattern is particularly evident. The head and the brain, it develops at much faster rate than the rest of the body. So that results in large head to the body, large head to the body ratio of the infant. After the birth, the cephalocaudal growth pattern continue while the neck and the upper body develop before the lower body and the legs. So that is why the infant, it can lift their head and sit up before they can crawl or walk. So the cephalocaudal growth, it is influenced by various factors like the hormones, nutrition, physical activity. So understanding the cephalocaudal growth pattern, it is important for the diagnosis and management of various disorders like the developmental delays and abnormalities. So remember, the head, it will take around, the head, it take 50% of the total length around third month of intrauterine life. So at the birth, the cranium, the skull is proportionally larger than the face. The skull, it is larger than the face. But the postnatally, the face grow more than the cranium. Remember, at the birth, the face is one eighth the bulk of head, whereas in adult the head forms 1/3 of the bulk. Now let us discuss what are the methods to study growth. The first are the longitudinal studies, other are cross sectional studies, and the third are the semi longitudinal studies. The semi-longitudinal studies, they are also known as hybrid longitudinal studies, as they are combination of the these are the combination of the cross-sectional and the longitudinal study. So number one are the longitudinal study. Longitudinal, the word itself means long. Long means following a group of people over a long period of time. Maybe when they are born until they are adults. So scientists, they can learn a lot from this study, but it is very difficult to keep people in study for so long. The second are the cross-sectional studies. That means looking at different group who are at different age at the same time. So scientists, they can learn a lot from this study, but they cannot see how individual they change over time. The third, they are the semi-longitudinal studies. They are the mix of first two. Now let us study the various methods to study the growth pattern. The first are the biometric. The biometric tests, they are measurement and evaluation of physical and biological characters like the height, weight, and bone density. These are done to determine the growth and development of the organism. Other method is use of implant. So what you do is you implant a small device like wire or marker into the bone or tissue at a particular location. So the growth of the surrounding bone or tissue, they can be measured over time by taking X-ray or other imaging studies. So the implants, they can be placed in the maxilla to study the growth in the specific area. The areas where we place implant, they are the hard palate behind the deciduous canine. So this area is studied prior to the eruption of the maxillary incisors, permanent maxillary incisors. Other is the below anterior nasal spine. This area is studied after the eruption of the maxillary incisors. The implant, it is placed below the anterior nasal spine, that is a bony projection in the midline of maxilla above the incisors. Other implant locations, two implants, they are placed at the zygomatic process of maxilla. Other is the implant, they can be placed at the border between the hard palate and alveolar process median to the first molar. In case of the mandible, The implant it is placed anterior aspect of symphysis in midline below the root tip. 2 pins on the right side of mandibular body, one under the first premolar and other below the second premolar of the first or the first molar. 1 pin on the external aspect of right ramus at the level of the occlusal surface of the molar. So these implant, their location they are chosen because of the precise measurement of the mandibular growth in the different regions. Other method we can use is the vital staining. So vital staining, it is a technique that is used in growth studies to label or mark the specific tissue in order to track their development over the time. So here we use is injection of the dye or tracer into the tissue. So as the tissue they will grow, the dye it will be incorporated in the new cells. So that allows the researcher to follow the path of development of the labeled tissue. So the vital staining it can be used to study the bone growth by injecting a dye into the bone tissue. So as the bone grows, the dye is taken up by new cells and it can be traced over time to measure the rate and pattern of the growth. Now what are the dye that we can use for the vital staining? The first is the tetracycline dye. So this tetracycline dye it will bind to the mineralized tissue and can be visualized under the UV light. Other dye is calcine. It can be visualized under the fluorescent microscopy. Other dye that we can use is the alizarin stain, alizarin red, xylenol orange that we can use. So what are the other things that we can use is radioisotopes. So the radioisotopes of the certain elements or compounds, when they are injected into the tissues, they incorporate into the developing bone and they act as a markers. So these radioisotope later be detected by tracking down the radioactivity they emit. So various radioisotope we can use is technetium, calcium, potassium, etc. Other method that we can use for estimating the growth is cephalometry. Cephalometry it is used to study the growth and development in the humans. So it determines the growth pattern. By cephalometry we can measure the growth pattern, evaluate the treatment outcome by measuring before and after treatment. We can determine the extent of improvement, whether the treatment was successful or not. Also we can study the genetic and environmental factors. Other we can use is hand, wrist X-rays. So it is a common site, the hand wrist, it is a common site for studying the growth and development in children, as the hand wrist it contains multiple growth plates or epiphyseal plate, so that are responsible for growth and development. So these epiphyseal plates, they are composed of cartilage that are replaced by bone. Other are the natural markers. Natural markers like the nutrient canal and trabeculae, they can be used to study the growth and development in the bones. The nutrient canals, they are the small opening in bone that allow blood vessels and nerve to enter and exit the bone. So trabeculae, they are the lattice-like structure within the bone that provides support and the strength. You should know the normal feature of the growth. One term is the growth normality, and other feature is variability. So normality, that means the normal feature of growth, they are the pattern of growth that occurs in the healthy individual that include the changes in height, weight and other physical character over time. So it is normal for the infant to double their weight by 6 months of age and triple their weight by 1 year of age. Variability fact that growth may that individual may growth at different rate, or they may follow a slight different pattern of growth than expected pattern. The factors that influence such conditions are the genetic, nutrition, physical activity, etc. Other important thing that you should know is what do you mean by differential growth? The differential growth, it refers to process by which different part or the tissue of organism, they grow at different rate or time. Like during the early stage of human development, we read that the head and brain, they grow at much faster rate than rest of body. So that results in large head to body ratio. Similarly, during adolescence, there is a growth spurt in which limbs and other part grow rapidly, while the torso, it grows more slowly. So that results in changes in the body proportion. So this differential growth, it is controlled by many factors like genetics, environment, hormones, nutrition, physical activity. So the differential growth, it is important aspect of development and have a significant effect on the form and the function of the organism. Now let us discuss about the growth concept. Now there are various ways by which growth occurs. The first is the displacement. So it can be either primary displacement or the secondary displacement. So displacement, it can be of two types. One is the primary displacement and other is the secondary displacement. So the primary displacement, it occurs as results of its own growth and remodeling process, while the secondary displacement, it results as results of external forces such as growth or the enlargement of the adjacent bone. The example of the secondary displacement, it is the forward displacement of the cranial base in response to growth and enlargement of the brain. As the brain grows and expand, it put pressure on the cranial base that causing them to shift in the forward and the downward position. So that can due to the alteration in the cranial base that can result in the shape alteration of the skull as well as facial features.